Well, it is a pleasure to be with you. I think my panelists are going to join me here momentarily, but again, at Georgia State is one of those interesting expanding footprints in downtown Atlanta, an institution that has now grown to more than 32,000 students with a very diverse health research portfolio that you'll hear a little bit more about as we introduce and have the panelists speak. I thought I'd just relate one very quick story to you, which as I was having a chance to meet folks in the lobby this morning, about a year and a half ago, maybe a little bit less than a year and a half ago, Russ Lapari and I were having early breakfast downtown. And Russ was going through essentially a discussion of what this conference was really shaping up to look like. And I said, Russ, one of the things that we should think about is really beginning to invite others into the conversation from beyond the Georgia, the Southeast area. And I'm delighted to report that a colleague that I saw this morning that I did not expect to see this morning has joined us from Australia, actually from Brisbane, Australia, who is here actually doing business at Emory. So we have now become international. I think it's a wonderful start. We at least have one individual in the audience who is representing an offshore location, and again, wonderful event. I do applaud the organizers in terms of what's been done. So if I could invite my panelists to join me, and I'm going to spend just a moment introducing each of them to try and conserve on our time. So please. So this panel is really about what is going on in the academy, what's going on in the university environment, and clearly we have some incredible research institutions that are in not just Metro Atlanta, but in Georgia. It is as someone who was a fairly recent arrival to Atlanta, being in Illinois for many years and then New York for a number of years. I am amazed at just what is available here as it relates to the resources, the infrastructure that is related not just to health research, but to many areas of research. And it's really gratifying, it's really heartening to see that we are beginning to kind of mobilize that effort in a very different way, which is wonderful. So I'm going to start at my far left, your far right. Dr. Michael Pierce is actually the director for the University of Georgia Cancer Center, obviously at UGA. Dr. T.J. Craddock is the director of the Protein Engineering Facility at Georgia Tech and Emory University. Dr. Richard Plemper, who is a professor at the Institute for Biomedical Sciences at Georgia State. And Dr. James Lillard, who is the Associate Dean for Research and the Director for the Office of Transformational Technologies at Morehouse School of Medicine. So, gentlemen, I'm going to join you, and then we will be off to the races. So, Mike, do you want to start and give us an opening on what is going on at UGA, and particularly as it relates to your science?
Great. Thanks very much. So I have a lot of hats at UGA. I do direct the Cancer Center, but I thought I would emphasize today our Complex Carbohydrate Research Center. That's where my laboratory is, and I'm principal investigator of the National Center for Biomedical Glycomics, an NIH-funded center to develop technologies to understand glycans and their functions. And the reason this is important is glycans cover all of our cells. So cells are sugar-coated, and each cell has a different type of complement of glycans on its surface. Even HIV is covered with glycans. So it's very important to understand the structures of those glycans, how they change, and how we can exploit that to develop disease markers, for example. So I believe that glycoscience is just poised to explode in terms of biotechnology funding and research. One reason is a good friend of mine, John Magnani, has a company who has a drug in phase 3 clinical trials as an anti-inflammatory with very low side effects. So I think this is going to be really one of the first big biotech drugs that are based on glycans. So at the CCRC, we have 18 faculty, 250 researchers in a building dedicated to glycoscience. And what do we need? We need collaborations. We have many collaborations across the US and around the world, but we need more collaborations because clearly glycoscience and glycans can impact all kinds of diseases, all kinds of biotechnology. So we need physicians, researchers, companies who have disease targets, who need markers for diseases. That's how we can help. We have collaborated with a monoclonal antibody company in Virginia, and with them we've developed an antibody that binds to a unique glycan on pancreatic cancer cells. So we have a very good marker now for pancreatic cancer, and I think that's going to move forward. So that's an example. And we also, of course, need entrepreneurs and we need capital to exploit the intellectual property that we've developed at the Complex Carbohydrate Research Center. For example, we have developed a new anti-TB drug that we have in mice now testing, but we need capital and we need folks interested in partnering with us to develop the intellectual property, not just from my laboratory, but from all the labs at the CCRC. So, thank you.
Thanks, Mike. TJ?
Georgia Tech's protein engineering facility and Gangbao's lab of nanomedicine are focused on curing diseases. And to do this, we're trying to develop nucleases, including CRISPR and tail nucleases, if you've heard them in the news lately. But it seems a little counterintuitive, even to me, that to correct a mutation, we want to cut the DNA. But that's what we want to do and take advantage of the cell's repair machinery and a good copy template that we throw into the cells. And thereby, we'll hope to correct genetic mutations and cure diseases. This is what we're trying to do primarily for sickle cell, where we cut inside the cells near the sickle cell mutation and supply a good copy of the beta globin gene. Same process for cystic fibrosis, and a few variants for things like HIV, where this is even in clinical trial with one of my former companies, where they're trying to mutate the HIV co-receptor, CCR5. In addition, we use these same gene editing technologies to aid research by making cell lines, or every kind of animal you can imagine at this point is a model animal for different types of research. Because it's a convergence of molecular biology, cell biology, computer programming, and all kinds of engineering to get these things into the cells, it's a very collaborative type of research, and we're very interested in expanding the number of cells, the number of animals, the number of different ways we've tried this. And so it's very important that we continue to do these things, because some of these nucleases were just developed in the last year, and we really need to see how far we can go to try and develop the last few steps to get these things into the clinic.
Thanks, TJ. Richard?
Yeah. Morning. So I am in the Institute for Biomedical Sciences at Georgia State, and the institute was really founded to foster translational research with a particular impact on human health. And so I joined the institute coming from the Emory Department of Pediatrics, and where I have built a research lab working on respiratory viruses, and in particular on the development of novel therapeutics against respiratory viruses. And so the institute really provided a very attractive home to continue this research while entertaining established collaborations with Emory Pediatrics, and we've heard about the Emory Institute for Drug Development. We also collaborate intensively with Dr. Painter and Dr. Liotta, and enjoy new collaborations at GSU. So I'm working on respiratory viruses, in particular a family of viruses that are some of the old pediatric pathogens, of which you probably have heard, like mumps and measles virus. But there are also newer or more recently discovered viruses of this family that are a severe threat to human health, create an urgent clinical need, and we, despite intensive research at the moment, do not have any effective vaccines. One of these viruses is respiratory syncytial virus, which is a major pediatric threat, in particular to prematurely born infants who are, if infected by RSV, who are in severe danger with very high mortality rates. And so we have recognized this as an urgent clinical need and have developed jointly with the EIDD several drug classes against different members of this virus family, some of which, the most advanced, are currently in the stage of licensing, or licensing is prepared for further development and ultimately clinical use.
Thank you, Richard. James?
Great. James Lillard, I'm at Morehouse School of Medicine. I was going to take a little bit different approach and give you a background of things going outside of my laboratory at Morehouse School of Medicine. I'm the director of our Office of Translational Technologies, and we're engaged in the entire documentation of the invention process and commercialization process. I guess we started about three to 4 years ago to do it in a more rigorous approach. Currently, we, because of our unique mission at Morehouse School of Medicine, focus on addressing or narrowing some of the gaps in health inequities. We have a number of research programs focused on neuroscience, HIV and AIDS, cancer biology, and others. I'm primarily a cancer biologist, and just to give you a little bit of background, our laboratory was among the first to use antibodies to target chemokines to address certain aspects of cancer metastasis. We now have some other biologics that are doing some of the same things, and I'm happy to be here.
Thank you, James. So let me start with again, incredibly diverse in terms of where you're staged as it relates to the continuum from discovery all the way to moving something into market, which actually does, at least for me, beg the question of the resources and the potential interactions or collaborations that would be really important in all of these different parts of the life cycle. So maybe take a minute or two and talk about what some of the really kind of critical resources, interactions, connections that would be important to really accelerate what you're doing at this particular moment in time. So TJ, let me start with you.
One of the things that we continue to find is that, to some degree, even though in some of the press these type of nucleases have been well documented, that I run into researchers who seem to find a roadblock. Oh, if I could only change this receptor to figure out if it's really where the virus binds, or if I could really figure out this question that the reviewers of my grants are giving me trouble about. And so I realized that there's people that actually have the need, but just don't know necessarily how easy it is now to take a cell line and change an individual nucleotide to change part of the protein. So we have this kind of need to tell people about our abilities these days and how the price has dropped dramatically. And to a lot of people, they know that there's companies offering this kind of process, but an academic lab, we can do it very inexpensive. And so we are trying to get more collaborators just to try and increase the numbers, as I mentioned, to understand better each of the elements. And so as we go through the process, so we have engineers inject these into cells, and we see how that changes the specificity. We continue to learn more, but we really have this need to try and have outreach or communication to get more people to try these so that we learn more about how to succeed in our primary target, sickle cell, or some of the other diseases that we work on. So we've got technical hurdles that I think will come from extending these kind of collaborations.
Richard, you're probably one of the closest in terms of really looking at a market opportunity in terms of your particular research program. Do you want to talk a little bit about what the challenge is of essentially going into market and what the potential, again, connects or the potential resources that would really be critical at this stage of your program?
Sure. So research in my laboratory, in particular early stage discovery, is predominantly supported by research grants from the NIH and from various institutes that are pursuing viral endpoints or infectious disease endpoints. And I think the NIH has really built a very strong program that is incredibly effective in initiating early research, early discovery, and the early stages of development of, for example, a screening hit that has potential, but is clearly not a drug yet, but maybe one day can be developed to become a drug. But where I really see where the challenge is this gap between advanced development, clinical trials, so large-scale pharmaceutical, and the mostly NIH, so grant-sponsored early discovery and early development. And in between, there is a real need for a solid set of tox data, of toxicity data, of increased numbers of animals in animal trials to really ensure efficacy of your candidate compound and also safety. And really this gap, this intermediate stage development, I see as one of the major challenges, in particular for academic labs, because this goes beyond the classical academic setup. It goes beyond the classical R01 type NIH funding mechanisms, but is not yet at that stage where large-scale corporate partners would become interested. And I think that many of very promising drug candidates in many areas, including ours, really are at risk of falling victim to this gap that stops development or stops early discovery of a compound is really not holding up, so let's drop it, let's move on, let's not waste any time and keep it alive for a long time. And I think this is a major challenge for translational development.
I absolutely agree. The so-called valley of death that so many very interesting technologies, intellectual property falls into and never does see the light of day again. So very well said. James, this is, again, an area that you have a lot of passion for and are doing a lot in it. Do you want to?
Yeah, absolutely. We have approximately 7 faculty startups currently that are in that gap we just talked about. We've been relatively successful with our SBIR, STTR grant funding that when we partner with a small business or a faculty startup where we absolutely have to effectively partner to move the technology forward. And I think even let's say the other 15% of the time when we engage a larger company for a technology, I think it's absolutely critical that we have to have the IND enabling type of data that's required that you just mentioned. And I think a key challenge in that is identifying some of the regulatory compliance expertise that's needed to better create or apply for funding, SBIR funding, STTR funding, to add that to the technology, increasing its value. That's one of our challenges.
Excellent. Mike, I think you said it very well that glycobiology is one of those hot areas in medicine right now and has the potential to really be transformative in so many spaces. Obviously, you've got a lot of things that are going on. Do you want to talk a little bit about what some of those resources might be that would really help to move what you're doing forward, not just individually, but as now a broader community? And as we both know, there are other universities in Georgia that are essentially collaborating with you in this space. This is really one.
I'm happy you asked me that. Yes, I mean CCRC UGA was one of the first centers to be set up to study glycoscience, and we're very happy to see investment being made at Georgia State, at Georgia Tech, and certainly Emory, my good colleague Rick Cummings there with a national center there as well. So I think the momentum is building. I would like to just make a comment that I think we're really losing a lot of really, really innovative ideas throughout all these areas, because there's really no mechanism to fund those now at NIH. NIH used to have R21s and things like that, which were for highly innovative ideas. But the R21s are now the old R01s, because when I review these, you have to have more or less an R01 type of background and preliminary data to get those funded. So to me, there is a huge gap at the present time. You can make some basic observations, and whether it's in glycoscience or anything else, but then how do you extend those observations? In the old days, there were mechanisms, but now, not so much. I mean, we actually started a seed fund in the Cancer Center, collaborations between GRU and UGA, and the administration has put some money into that just to fund the types of innovative ideas that could really be transformative, that could really probably get funded someday. So I see that. I just wanted to mention I really see that as a possibility. As far as the glycoscience goes, it's so broad, everything from vaccines to anti-TB to modulating the immune system. I mean, those sugars are out there modulating really all aspects of the immune system. The IgGs that biotech companies make have to be glycosylated correctly for them to be active. Amgen, who I'm really familiar with, they patented a new glycoform of erythropoietin, and that's their lead drug now. And it all has to do with having the right glycans, because that regulates the half-life in serum. So it's a very, very broad area, and I would say every day someone has, at the CCRC, someone has a really great idea that gets written down and sent in to our tech transfer office as being something that could really make an impact.
So, Mike, you brought up NIH, and I think it's a good thing for at least the rest of the panelists to comment on, because I do believe that we are in this moment in time when either we're going to continue to leverage and take advantage of all the innovation that has happened, or we are seriously gonna step backwards, and that's a very troubling thought. An NIH budget that was at $32 billion, now going down to somewhere in the neighborhood of 29. Sounds like an awful lot of money, but when you look at just the way that is spread across the entire research environment, at least in the U.S., it's incredibly, incredibly competitive to essentially stay part of that NIH group of investigators. Richard, let me ask you. So you have had a fair amount of success in this space. You continue to be very well funded. What do you see as the landscape? Are there other opportunities out there that we should be thinking about beyond the federal government to really help support what is going on in labs like yours?
Yeah, let me just follow up briefly on the situation at NIH. I mean, I really think that the mechanisms in place, the system that is built, the review system, I think is outstanding. In my opinion, is unparalleled globally. But having said that, I mean, at the moment, so I'm sure we all review a lot of applications, go to study sections, and it is really heartbreaking to go to listen to discussions, to actively discuss proposals that are highly innovative, that have really very strong merit, potentially make a major impact, and we all know that essentially there are no funds to support what is meaningful science. So it's not the bad proposals that don't get funded. I think it's really a lot of very strong, very good proposals are just not making the cut. So having said that, I mean, increasingly we see that other resources are approached, foundation funding. NSF is another strong, major funding organization. But the problem really is that that I think there is no source that is really flush to the degree that could compete with NIH funding at the level a few years ago. So I think we just face this as a reality. There are alternatives, but we will face as a reality that that funding is very tight, and that globally we are, what biomedical research is concerned, we are clearly at risk of falling behind.
Yeah, just to echo that, we've been relatively fortunate at Morehouse School of Medicine. Our funding rates are still highly competitive, typically above what you see at institution per institution. I think that has to do with the significance or impact of the type of research, and that's aligned with our mission of serving unmet medical needs in underserved and rural communities. We've also began to apply for more SBIR, STTR funding. I should point out before I forget, the Georgia Research Alliance Venture Lab and Venture Program has been extremely helpful to us in a number of our startups, again, bridging that gap. But again, there are limited funds. So what we've done pretty effectively, we've begun to collaborate with groups in China, in the UK, and to help bridge that gap, and it's been effective so far.
Excellent. Mike, anything more that you want to add to this, or did you already cover it?
I think we've covered that.
TJ, anything?
I have a couple examples that are central to our research that exemplify how basic science is important even to translational science, and that the two nucleases that I've discussed were first observed in plants and bacteria. So if there wasn't the basic research on plants and bacteria, we wouldn't have learned about these nucleases that are headed into translation into the clinic. So when we think about defunding or putting less money into basic research, it really does have long-term implications. So we're hoping that things will improve.
And I think we are, at least I am very optimistic. There are a number of different groups that are now looking at really expanding, whether it's the Angel Network or whether it is the seed environment, whether it is the early professional rounds in the private equity venture space, but really looking at how more resources can essentially be put into the pipeline to indeed ensure that we are at least post-discovery, that we have some capacity to continue to move that forward. The challenges exist, as you've all said, from literally the discovery phase through translation and into market. So there is no easy fix in any one of these in any one of these spaces. We've got just a couple of minutes left. I'd like to just give you all 30 seconds to essentially any other topic that you want to essentially cover quickly. Would like to give you the opportunity to do that. So, Mike, let me start with you. Anything that we've not touched on that you'd like to?
Well, I'd just like to make the comment that even though we may sound like we're a little pessimistic here, I mean, we are planning for the future. We have a new training grant at UGA to train the glycoscientists of the future. That's really going to help us build our infrastructure. So this would be for graduate students, and we're going to recruit all across the state of Georgia and the Southeast to bring in qualified graduate students to train them in glycoscience, because we really do see this as a huge growth area for biomedical research in the future, and so does NIH by giving us the grant.
TJ?
I just have one personal note, having gone to school and worked in Boston and the Bay Area. I was really pleasantly surprised when I moved to Atlanta and started working at Tech that the amount of small biotech, the amount of collaboration between the different universities, and the number of projects that we have that really involve a lot of the resources we have here. So I was, again, pleasantly surprised when I moved here, and even though my folks still live here and I come here quite often, just the level of all of that.
Incredible resources. Richard?
I'd like to second that. I think translational research is really an interdisciplinary effort, and the Atlanta area, or the Georgia at large, I think with the density of outstanding universities, of outstanding groups, I think is really the perfect basis for productive and fruitful collaborations. And I think this meeting, this conference, is a prime example that collaborations are actively supported.
Well said, James.
In closing up, I think it would be wonderful. We have a kind of a blanket collaborative research and development agreement with Emory University. I'd like to propose that I guess all of our institutions could do something like that to kind of grease the skid, so to speak, to not get in the way of innovative research. So maybe we could take that on.
A great note to end on. So please help me thank these distinguished scientists. Very, very well done, gentlemen.